last one in the current series. But the slides and video archives of all the lectures, including this one today, will stay on OEHA's website for all of us to continue to learn. In addition, we have a journal club on genetics and epigenetics. The first journal club meeting took place last week, July the 15th. It was very good. And if you want to continue to hear about the latest findings on genetics and epigenetics, and want to learn how to put those findings into our daily work of risk assessment, risk management, please join us. And I want to use this opportunity to thank everybody. I'm not going to read all the names on this slide, but I want to stress this series is really a product of teamwork. Many people have worked on this directly or indirectly. I'm really grateful to everyone, and including all of you who had made the effort to come into the room out of your cubicle and to watch our lectures on the web. And that's all I want to say. Let's welcome Dr. George Alexeyev, OEHA Scientific Director, on behalf of Director John Denton.
Good morning, everyone. It's my pleasure to introduce the speaker this morning. Dr. Prins is Professor of Physiology in the Departments of Urology and Physiology and Biophysics at the University of Illinois in Chicago. She has also been director of the University Andrology Lab at the University of Illinois since 1996. Dr. Prins was president of the American Society of Andrology from 2003 to 2004, and she has been on numerous distinguished scientific panels, such as the NIEHS/EPA Expert Panel on Bisphenol A in 2006, and the NIEHS External Scientific Review Committee since 2004. Dr. Prins received numerous honors and awards for outstanding research, has published more than 125 peer-reviewed papers. She has many research programs at her laboratory. One of them focuses on hormonal regulation of the prostate gland growth and carcinogenesis. Dr. Prins' research on the prostate has significant impact for society, particularly since prostate cancer is the most common non-skin cancer in the U.S. It affects 1 in 6 men. Today, Dr. Prins will present her findings that indicate early life exposure to natural, synthetic, or environmental estrogens, such as low levels of bisphenol A, increased susceptibility to precancerous prostate gland lesions with aging. Dr. Prins will discuss recent data suggesting the neonatal estrogen reprogramming may promote prostate gland disease in aging men through epigenomic alterations during development. So thank you very much. Let us welcome Dr. Prins. Are we using... Okay, this one, sorry.
All right, thank you very much for that wonderful introduction. It was very succinct and very precise. And so I'm excited to be here and to do a webinar broadcast. I've never done one of those before outside of the immediate building, and so this is a new one for me. And so I'm going to talk about my work for the past 15 or 20 years, actually more than 20 years, but we'll not go there, on hormonal regulation of prostate growth. So by training, I'm a physiologist, and my research is always centered on my basic research on prostate gland. And so I'm very centered on the prostate gland, and that's what I'll be talking with you today with regards to what some estrogenic components might be doing to the prostate. So let's see if I get this right. So it's not advancing. There it is. So realizing that I'm talking to an audience that might not be as familiar with prostate cancer, I just have a slide here to talk about prostate cancer and the magnitude of the problem of this disease. As was said, it's the most prevalent non-cutaneous cancer in American males. In the latest statistics, 2008 statistics, show there are about 200,000 new cases annually, and about 27,000 men died of prostate cancer in the United States in 2006. It's the second leading cause of cancer deaths in U.S. males, second only to lung cancer, and it's of similar magnitude to deaths from breast cancer and HIV in the United States. There are a few risk factors for prostate cancer that are known and established, and one of them is ethnicity. There's a 2-to-1 incidence ratio in African American males versus Caucasian males in the United States, and I'll say that more on that in a moment with regards to estrogen. Asian males have the lowest incidence, and this is not only in the United States but also worldwide. There's also the other major risk factor is age. So, of course, age-related incidence, up to 30% of men over the age of 50 harbor silent prostate cancer. And the percentage of men in the United States who will develop invasive prostate cancer with age increases. And there will be a lifetime, let's see if I get this right. No, that was the wrong button. Oh, that was this. It's like a Wii, which I've never used. Okay, anyway, birth to death, you can see it, 1 to 6 chance of having invasive prostate cancer. All right. So research in my laboratory has focused on prostate development. So I want to take you away from aging and look at the other side of the spectrum, the time in life when the prostate gland develops. Now, a few things to say about the prostate gland. The prostate is one of the accessory sex glands in the male. It produces a component of the seminal plasma. Most of the male accessory sex glands arise embryologically, so these would include the seminal vesicles, this little oh, there it is, the seminal vesicles, the vas deferens, epididymis, Wolffian ducts. Most of the accessory sex glands in the male arise from the Wolffian duct, and the Wolffian duct is a mesodermal structure. The prostate is unique in that it's the only accessory sex gland that arises embryologically from the urogenital sinus. It's an endodermal structure. And there's been talk for decades that this unique embryologic origin of the prostate gland has a basis in why this gland has such a high incidence of cancer. You don't hear of cancers of the seminal vesicle. I don't think there's ever been a reported case of cancer of the epididymis. And so these other organs have very low rate cancers. The prostate, coming from the urogenital sinus, has a very high rate of cancer. And so its embryonic origin appears to contribute to cancer of this structure. Now, in the human fetus, this is when prostate development occurs. It occurs entirely in utero, and it's initiated at about the 10th to 12th week of gestation in response to testosterone being produced by the fetal testes. And so testosterone levels start to rise, and they start driving the morphogenesis of these other reproductive tract organs. Now, androgen levels peak in the second trimester in the male and then begin to decline into the third trimester. And so it's during this period of time that we have a lot of growth of the androgen-dependent male accessory sex glands and their morphogenesis. Now, during the third trimester, in addition to androgen levels declining, estrogen levels from the mother begin to increase, and they reach peak levels in the third trimester. Now, humans are normally protected by maternal estrogen from alpha-fetoprotein, which binds to the E2 and protects the male. But there's clear evidence that some of this estrogen, elevated estrogen, is getting through the male. All the males have squamous metaplasia in their prostates at the time of birth, and this is immediately sloughed at birth when the estrogen levels fall. And so there's clear evidence that maternal estrogens do have a direct effect on the developing prostate gland. In fact, as far as estrogenic effects go, the developing prostate gland is the most sensitive period in life. We now know this is becoming a very hot area of research, is the role of estrogens in the prostate gland throughout life. But relative to the estrogenic effects in the adult animal, the estrogenic effects in the neonatal or fetal human are much, much higher. And so I want to just introduce to you a paradigm called the developmental origins of adult disease, or the DOHAD of human disease. And this regards developmental programming or reprogramming. And it's believed, this hypothesis, and this is not just for reproductive tract animal tissues, but for a lot of tissues in the human, that there's critical periods or windows in fetal or neonatal development when stimuli have long-lasting, i.e. life-spanning effects. And if we look at a model for what's going on in the prostate gland with regards to rapid growing periods of time, seen here in blue, the rapid growing growth periods are during the fetal development, during fetal development, during puberty when the prostate grows, and in disease, in aging when the prostate grows, be it BPH, benign prostatic hyperplasia, or prostatic carcinoma. These are the rapid periods of growth. Otherwise, the prostate's relatively quiescent. And I'm going to show you evidence from my lab and others that hormones can imprint or reprogram the prostate gland during this developmental period, which have long-ranging effects affecting the disease susceptibility in the adult. So what are some of the developmental estrogenic exposures that we might be concerned with regards to humans? Well, in humans, we have elevated maternal estrogen levels. There's a lot of evidence that I just spoke to you about how maternal estrogens do affect the prostate gland, but African American women, for example, have 40% higher E2 levels during early pregnancy as compared to their Caucasian counterparts. And this was shown by Henderson and Ron Ross and that group down at USC to very tightly correlate with a higher incidence of prostate cancer in these patients, in this population. And they've linked this with some polymorphisms in steroid metabolizing enzymes. There's other evidence as well. Indicators of pregnancy estradiol levels have shown a significant correlation between elevated estradiol levels in the mother during pregnancy and prostate cancer risk in the sons. The largest evidence, however, comes from the DES exposure. And I'm sure everyone here is familiar with the DES. It was taken by pregnant women between 1950 and 75 to prevent spontaneous abortions. It increases vaginal clear cell carcinoma in the DES-exposed daughters, and this was discovered by Arthur Herbst at the University of Chicago, and from that, its use was discontinued. Now, when we look at the sons, at their prostate glands, there is evidence that early on their prostates show permanent alterations. But this really hasn't been followed up extensively, the DS-exposed sons. If we look at rodent models which accurately predict the female lesions, we find that DS exposure early on does predispose to prostatic tumors. And then there's the endocrine disrupting chemicals. Methoxychloroquine and bisphenol A were shown to, developmental exposure was shown to affect prostate growth in some strains of mice. And for some of these chemicals, there's concern about bioaccumulation. And so this is really the rising interest out there in the public and also in the scientific community about endocrine disrupting chemicals. So I just want to say a few things about BPA for those who aren't completely familiar with it, and if you are, just bear with me. So the sources of bisphenol A in the environment are largely from polycarbonate plastics. Polycarbonate plastics are a long chain polymer of bisphenol A monomers. It's also in dental sealants and epoxy resins, which line the cans of most of our tin cans. And for the most part, these long chains of polycarbonates are relatively stable molecules. And they should not be affecting us. But in fact, BPA monomers are released upon heating, where we have hydrolysis of the ester bonds between the long chains of the polycarbonate polymers, releasing BPA, which is highly water-soluble, into the immediate fluid or food environment. So to just say it very quickly, this is how BPA is getting into our water sources and the environment, and this is whereby humans might be exposed to it. Now, interest in BPA really began to rise when the CDC released a report showing that 93% of humans that were tested had BPA metabolites in the urine. And as was stated, I was on this BPA panel by the NIEHS last year, or 2 years ago now, and we discussed this. And since BPA is such a rapidly metabolized compound, to have this many people having it present in their urine at any given time suggests that there might be chronic exposure to BPA, and that it's somewhat similar amongst the population within people. It doesn't vary as much as one would initially have suspected. The unconjugated BPA in human serum that's been reported has been highly variable, but it's in approximately the 0.3 to 5 nanogram per mil range. It's present in the fluids that the developing fetus is exposed to, and suggests that the developing human fetus is chronically exposed to BPA. And so there's concern about early life exposures to this substance. Now, I was the chair of the BPA of the NIEHS expert panel on BPA. I was the chair on the sub-panel of looking at the carcinogenic activity of BPA, and the results of this were published in Reproductive Toxicology. And so this was a group of 37 scientists who have done work in BPA. And there was a group of these scientists who have expertise in cancer research. And it was our charge to look at all the papers where there was any effect or investigation of BPA as it relates to a carcinogenic situation. We had to look at all that and write a review saying this is what is certain, this is what is suggested, and this is what's uncertain. And this, I just want to share with you our conclusions. This is what is certain. We know that there is reproductive toxicology due to early life exposure to diethylstilbestrol. That's absolutely certain. That prenatal DES exposure increases risk of breast cancer and uterine fibroids in women and animal models. That estrogen is a known carcinogen, and that BPA acts as an endocrine disruptor with estrogenic properties, among others. What is suggested by the literature is that, and this comes from an NTP study done by the agency themselves in 1982, that high doses, very high doses of BPA increased hemopoietic cancers and Leydig cell tumors in rodents, suggestive of carcinogenesis. Now, this would not be at normal human exposure levels. Early life exposure to low-dose BPA may induce or predispose to preneoplastic lesions of the mammary gland and prostate in adult rodents. BPA alters microtubule function and can induce aneuploidy in some cells and tissues. This was really coming from the work of Pat Hunt with the oocytes at environmentally relevant doses. BPA may induce cellular transformation in vitro. What was particularly interesting to me as well is a study from Karen Knudsen's lab that BPA can ligand to mutations of the androgen receptor that are commonly found in advanced prostate cancer. And when it ligands to them, they can activate the androgen receptor. And so men with prostate cancer who are on hormone deprivation therapy can have their androgen receptor in their prostate cancer activated by environmentally relevant levels of BPA in their serum. So there's a concern there. What is uncertain at this point is whether developmental BPA exposures increases mammary or prostate carcinogenic risks in humans, whether BPA exposure directly or indirectly induces or promotes cancers in mammary and prostate, whether BPA increases cancer susceptibility in all estrogen target organs, and certainly what's uncertain is its mode of action. Is there an estrogenic pathway involved? Is it a non-estrogenic pathway? Are epigenetics involved? And these are questions for the future. Now, there at the same time was another panel of scientists who was convening and looking at pretty much the same literature. And this was run by the NTP. Now, this was another expert panel. These were chosen by not having worked specifically in bisphenol A in their own research in order to limit bias from that level. And their charge was very different than the charge of the NIEHS EPA panel, and it was to look at the level of concern for humans in the population. And the level of concern that the NTP comes up with are on a scale of 5 over there, with concern being the highest level and negligible concern being the lowest level. And the highest level that they came up with in their draft report going forward, which has not yet been finalized, is some concern. And some concern was found for neural and behavioral effects of BPA in fetuses, infants, and children at current human exposures. There is some concern, they determined, for BPA exposures in fetus, infants, and children at current human exposures based on the effects in the prostate gland. So this was highly controversial. The panel itself initially recommended minimal concern. This was sent on to the NTP, which raised the bar based on public comments and some additional data for the prostate gland from minimal concern up to some concern. And to a certain degree, that was based on work that I'm going to show you today from my laboratory. There's minimal concern based on the effects in the mammary gland in early puberty. The NTP actually had recommended raising the bar from minimal to some, and then most recently in June, the Board of Scientific Counselors bumped that back down to a minimal concern level. So this is the latest in terms of that concern. And then there's negligible concerns with regards to pregnant women, birth defects, decreased birth weight, and so on. And one can find this by going to the NTP website. Okay, so I want to show you the data for the prostate gland. So in contrast to the human, the rat prostate development is initiated relatively late in life. And so we start seeing testosterone being formed in about 14 to 15 days of life. This is with a 21-day gestation period. And so the prostate gland initiates just about 2 days before birth. And at the time of birth, the prostate gland, this is the rat Sprague-Dawley prostate. Actually, all the rat prostates look like this. The mouse is very similar. Is very rudimentary at the time of birth. And it has large branching morphogenesis that occurs during the first 5 to 15 days of life. Okay? And what develops is a lobe structure in the rodent where we have several different lobes. So this is not like the human prostate in that regard, where we have the ventral prostate, the lateral prostate, and the dorsal prostate. I can't point them out there. I can't work this. But we have this prostate gland that has a lot of heterogeneity, and it's a good model with some caveats for the human prostate and human prostate disease. Okay? And so work by Howard Byrne and McLaughlin in the 70s, Coffey in the 80s, and myself since the early 90s has looked at this phenomenon of a developmental estrogenization. That's what I've always referred to it as. You can also call it estrogen reprogramming of the prostate gland. And in this model, we have a brief exposure to estradiol on day 1, 3, and 5 of life when that morphogenesis is occurring. And then we lay off of it, let the animals live happily in their cages for a while. And as they age, we start seeing lobe-specific developmental and differentiation defects. And in adulthood, with aging, they start to see increased incidences of dysplasia and tumor formation. And so here we see adenomas throughout the ventral prostate gland. We see high-grade prostate intraepithelial neoplasia, the precursor lesion to prostate cancer, a lot of squamous metaplasia and inflammatory cell infiltration. Now, this is work from my laboratory using high doses of estradiol, and it was a model for DES, for pharmaceutical exposures. And so we wanted to understand the mechanisms for this. We've published a lot and done a lot of research on a number of molecules that are affected. Believe me, there are a lot of molecules that aren't affected, and I'm just showing you a summary of some of those findings up here on the slide. But I just show it to demonstrate to you. Oh, now I missed a model. Oh, okay. Well, it's maybe the slide's not there. Yeah, the slide's not there. And so what the slide would show was that we believe that what's happening with estrogen is that normally you have an undifferentiated epithelial cell in the early newborn prostate, and it gets differentiation programs from testosterone through the androgen receptor and becomes a functional differentiated epithelial cell. So that's a normal process for epithelial differentiation. And if during that early period of time, those cells also have estrogen receptors, if we see an abnormal exposure to estrogen at that cell, it induces permanent differentiation defects in those cells that now sit in the prostate gland and serve as precursor cells for dysplasia in the aging animal. So that's what that model would show. So more recently, we asked the question about dose response. Okay, that was at high doses. What about low doses? And the reason this question came up is some very controversial work from Fred von Sahl's group in 1997, which said that there was a low-dose, non-monotonic effect of estrogens on the prostate gland. And so here we're looking at diethylstilbestrol, and what they found out was at very low doses, is that instead of suppressing the prostate gland, as we see at high doses, you actually stimulate the growth of the prostate gland. And he also found this for bisphenol A. And so he called this the inverted U-shape curve and said that there were different responses of estrogens and that the low doses of estrogens may be problematic. And so there are several questions that have been asked over the time since then. Does a low-dose effect really exist? So that's certainly been batted around. Is it reproducible? Are there species or even strain differences? And I think the work from Jimmy Spiro really points towards that. Question that I asked as a prostate biologist and a cancer researcher was, does it drive prostate pathology? So in the tissues that were shown here from von Sahl, where you saw bigger prostate glands, they were all histologically normal. They were just bigger. And so the question is that problematic? Is that going to result in more disease with aging? And that's what I wanted to know. And so we did a dose-response profile in our Sprague-Dawley rats over a 7-fold log dose of estradiol itself. And what we found in our Sprague-Dawley rat, as well as in the Fisher 344 rat, which is more estrogen sensitive, is that there was not a low-dose stimulatory effect that was statistically significant in the adult animal. And when we looked at the histology, this differentiation of the cells also appeared to be normal in the low-dose estradiol groups. Now, we did see low-dose estrogenic effects on the testes. We just didn't see them coming through on the prostate gland. So, great. My next model, see, my model slides are coming through. Okay, so this is what the model slide was going to show you. We asked a different question. What we asked next was, okay, maybe the low doses of estrogen don't directly drive prostate pathology. We wanted to know if they could alter the susceptibility of the prostate gland to a carcinogenic agent. All right? And so estrogen is a known carcinogenic agent, and there's a wonderful model for hormonal carcinogenesis in the rodent animals, and it's a good model for human cancer as well, where in the adult prostate, in the adult rat, the noble rat specifically, you give elevated estradiol, about 5-fold elevated normal levels of estradiol, and you do that for 16 weeks. Clamping the testosterone levels so they stay constant, and you'll drive high-grade pin lesions in 100% of the animals. So there's adult estrogen-induced carcinogenesis. That's in the noble rat. in the Sprague-Dawley rat, which is less sensitive to estradiol, they have a much lower incidence of estrogen-induced carcinogenesis. So we asked, well, what if they saw a hit? We asked a two-hit type question. What if they saw an exposure to BPA or low-dose estradiol early on in life? Would those Sprague-Dawley rats be more susceptible to prostate lesions in adulthood? And so we set up this two-hit model in our Sprague-Dawley rats. And so the first hit that we gave was at day 1, 3, and 5 of life, either oil as a control, a high dose, because we know that works, a low dose of estradiol, and this is the low dose that does cause testicular effects, and a low dose of bisphenol A at 10 micrograms per kg body weight, producing a predicted serum unconjugated BPA level similar to human exposures. We then allowed these animals to go to day 90, and group were divided in half. Half of them received a testosterone plus estrogen pellet for 16 weeks, and the other half received empty capsules. And then at the 7 months of age, we sacrificed the animals, collected all their tissues, and we did histologic analysis blinded. So it was a blinded analysis for treatment groups. And this is what we found. I'm going to show you the data from the dorsal prostate, and I'll admit it's because that was the cleanest looking data. The ventral and lateral prostate data are online on a supplement in Cancer Research. All right, so our animals at 7 months of age that had seen oil neonatally looked normal. They had a very low PIN score, very low prostate intraepithelial neoplasia incidence. If they had seen high-dose estradiol neonatally, they had a very high PIN score and a nice high PIN incidence at 7 months of age. If they'd seen low-dose estradiol neonatally, they also showed an elevation in the PIN score. So we were surprised at that. We didn't see that at day 90, but now it was coming through at day 200. If they saw bisphenol A neonatally, in adulthood, they did not have statistically elevated PIN score, and pretty much all the prostates looked normal. What happens when we now give this second insult to the animals? Well, our oil-treated animals, similar to the literature, showed an increase in PIN scores if they were treated with oil neonatally in just the adult T plus E. And about 40% of the animals showed a mixture of low-grade and high-grade prostate lesions. If they had neonatally high-dose estrogen plus elevated T plus E, they really developed PIN high-grade in all of the animals. If they saw low-dose estradiol neonatally and then adult estradiol, the PIN scores were no different than if they had seen oil neonatally. So there was no additive or synergistic effect whatsoever. However, if they saw bisphenol A neonatally and then adult estradiol, 100% of the animals developed high-grade PIN lesions. We then looked at the proliferation indices and the apoptosis indices, and once again, these were, which are normally very low in a prostate gland, these were elevated only in the high-dose estrogen groups and without adult exposure, and high-dose estrogen with adult exposure, and BPA with adult exposure. So altogether, this suggests that this developmental exposure to estrogens initiates or activates precancerous pathways, because abnormal proliferation and apoptosis rates are markers for an imbalance in cell proliferation and apoptosis, which are thought to contribute to prostate pathology with aging. So to just summarize, we have evidence that a range of estradiol exposures from low dose or biologically elevated levels to a high dose pharmacologic exposure can predispose the prostate to PIN lesions with aging. That neonatal exposure to environmentally relevant doses of BPA may increase the susceptibility of the prostate to carcinogenesis with additional adult insults. And I just want to mention that in aging men, their estradiol levels rise. This isn't just a weird animal model. It has biologic relevance. Testosterone levels begin to decline, and relative estradiol levels rise, not only because of the decline in testosterone, but you get a lot of production of estradiol from the adipose cells. So, of course, the big question is, how does a brief low-dose exposure permanently alter the memory of the prostate gland, that it remembers that exposure and then responds differently in adulthood when it sees that second hit? And I just want to put in this slide about estrogen action. So I don't want to talk about direct estrogen effects on DNA mutations, et cetera. I want to talk about estrogen action through the estrogen receptor. It binds to the receptor. And most people, when they think about it, think about activational responses that one typically sees in an adult. These are reversible effects. The response lasts as long as the estrogen is around. So, for example, the cyclic changes in uterine receptivity. You remove the estrogen, the effect stops. Okay? But it's also long been known, particularly in the brain, that there are organizational actions of estrogens during the developmental period, and that these effects of estrogens are irreversible. And they're irreversible due to changes in organization during the developmental period. Structural organization of the tissues or cells due to direct estrogen action on gene transcription, on proliferation rates, etc., within those cells, directing them into certain sizes, certain differentiation programs, certain neural circuitries. There's also epigenomic organization that's taking place at this time, DNA and chromatin modifications, which organize the genetic information. And so there's evidence that estrogens can have irreversible actions when they're exposed during these developmental critical periods of time. And so we turned our attention to looking at some epigenetic modifications. So you're all familiar with that. You're having a nice series on that, which are mitotically heritable alterations that are passed on from cell division to cell division. And there's three basic types, and we're going to focus today on DNA methylation. And the reason we came up with this hypothesis that estrogen reprogramming of the prostate may be mediated in part by some of the epigenetic alterations due to changes in methylation patterns of prostatic genes. And the reason we came up with this was a publication by McLaughlin 10 years ago showing that there was abnormal demethylation of a particular CPG site in the lactoferrin promoter of the uterus of DES-exposed mice, and that it was correlated tightly with the DES effects in the uterus. So that's why we decided to initially focus on DNA methylation. And I don't really need to go through gene methylation and DNA methylation very much. You're aware that methyl groups form at CPG dinucleotides, which are found as aggregate CPG islands. The whole process is catalyzed by DNA methyltransferases, and which add the methyl groups, and then they recruit in methylated DNA binding proteins, which have a number of effects besides just blocking transcription of binding factors. Importantly, DNA methylation has also been identified as a very important process in cancer, perhaps even more so than genetic modifications in cancer. And so they've been shown to contribute to initiation and promotion of all kinds of cancers, including prostate cancers, and it can be due either to hypo or hypermethylation of the DNA. One can have global hypomethylation, which leads to chromosomal instability, and one can also have gene-specific hypo or hypermethylation, which leads to inappropriate gene transcriptional activation. And so you can think if you hypermethylate a tumor suppressor gene, that's going to be bad. And if you hypomethylate and allow elevated expression of normally suppressed oncogenes, that's likewise going to have detrimental effects. So this was done in conjunction with Shuk-Mei Ho. I believe she already showed you some of this data in the first lecture because I saw it on the posted stuff. But just to review, this was our experimental workflow. We took all of our estrogenized tissues, we extracted the DNA, and then we interrogated them for changes in DNA methylation using methylation-sensitive restriction fingerprinting. And when we found interesting differences between treatment groups, we looked at those and followed those leads up. And so the approach that we used was methylation-sensitive restriction fingerprinting, where one just takes all the genomic DNA, which normally would look like this, and we treat it with MSE1 digest, which digests the DNA into pieces at TTAA sites. Okay, so now we have just a random amount of sites, smaller pieces of DNA. we now treat it with an enzyme that's sensitive to the methylation status of that DNA. And if it's non-methylated, it gets chewed up. But if it's methylated, it remains. And then with random primers, we PCR up the products. And we'll get products wherever there was a lot of DNA methylation. And the beauty of this approach is that it allows for global methylation pattern screens. So we're going to look at changes, period. I'm not going through and looking at a gene that I previously identified as being interested, such as maybe some of the Hox genes, et cetera. I just want to see what's out there. And so in our initial screen, we came up with over 50 candidate genes, and a lot of them turned out to encode for signal transduction molecules. And we've at first followed up phosphodiesterase type 4, variant 4, which I'll talk about to you today. So this is an enzyme which is critical in all cells where it's expressed in that it breaks down cyclic AMP. And cyclic AMP is known to be very, very important in all kinds of processes within the cell, including cell cycle progression and apoptosis. And importantly, it's been shown to be involved in tumor cell growth in a number of tumors, and it's being used right now, looked at as potentially a therapeutic site for therapeutic intervention. So we picked this particular one out because the site that we found that was showing methylation differences following estrogen exposure was in the 5' region, which suggested that it might be a regulatory region up here. And so we performed a 5' race, identified the transcription start site, et cetera, and all the different important elements in the rat PDE4. And we found a large CPG island, which would be a potential methylation site in the promoter 1 first intron of this gene. And so we performed bisulfite genomic sequencing, and for the most part, all of these sites are non-methylated. That would be the open dots, except one site which is consistently methylated right prior to the translation start site. But what we found was this is an example of one of the treated animals, either with oil or bisphenol A. As the animals aged to day 200, we started finding this loci down within the first exon that started becoming hypermethylated. And interestingly, it didn't become hypermethylated if the animals had been treated with any of the estrogens neonatally. So we interrogated this more clearly using methylation-specific PCR, working with a primer that primed up just that region. And the primers primed up either methylated or unmethylated sequences. So let's look at the oil-treated animals here. At day 10 of life, there was an equal distribution of methylated and unmethylated sequences at that loci. But as the animals aged, by day 90, that's young adulthood, they became mostly methylated. It became hypermethylated. And this was associated by PCR with a decrease in expression of this gene. But what happened if any of these animals had been exposed to either high dose, low dose, or BPA? As they aged, instead of becoming hypermethylated, this gene now became hypomethylated. And this was associated with elevated expression levels of this gene throughout life. So we're now tying this in with alterations in gene transcription. So to look at this more closely, Winnie Tang in Chukmei Ho's laboratory is doing some work with rat epithelial cell lines where she can look at things more directly. And there are two cell lines, a normal rat epithelial cell line, the NBE1, and a tumorigenic, and they both come from the noble rat. And so they first looked at the methylation status at that region in the normal epithelial cell and found that it was hypermethylated, whereas in the tumorigenic cell, it wasn't hypermethylated at several of the sites. So that mimics what we're seeing in the estrogenized prostate. Secondly, if you treat the cells with the demethylating agent, 5-azideoxycytidine, and remove these methyl groups, you can increase expression in the normal epithelial cells. You don't in the tumorigenic cells because they're already hypomethylated. So this shows that these tumorigenic cells may be a good model for the estrogenized prostate, and that this appears to be a methylation-regulated gene. So some more recent work that was just presented at the AACR meetings looks at the direct effects of BPA and estradiol on these epithelial cells in culture. So here we're looking at PDE4 expression in the NBE cells that are exposed to bisphenol A or exposed to increasing concentrations of estradiol. And what we found was that with increasing concentrations of BPA or estradiol, you could increase the expression of phosphodiesterase 4. Not in the tumorigenic cells, however, but in the normal cells. And this was directly correlated with an increase in hypomethylation of that gene, very similar to what we found in our animal model, both in response to BPA or to estradiol. So BPA and E2 do directly increase PDE4-D4 expression through gene hypomethylation in normal cells, but not tumorigenic cells. Furthermore, this effect was blocked by an estrogen receptor antagonist, an ICI compound. These cells have some ER alpha, but they mostly express ER beta. So we wanted to find out, well, how is estrogen doing this to these genes? So we looked at a number of molecules involved in this process. We looked at the DNA methylation transfer enzymes, DNMT, the ones that do repair methylation or de novo methylation in the cells. And we also looked at some of the methylated binding proteins. And for the most part, there were some differences I won't go into. None of the DMNTs were different with BPA or estradiol. But the one that was most interesting with regards to methylated cytosine binding proteins was MBD2. Now, interestingly enough, MBD2 is not only is recruited to the complex when there's a lot of methylation at that site, but there's evidence now that it's a putative DNA demethylase and that it may demethylate at specific sites, forming an internal negative feedback loop with regards to methylation regulation. And what Winnie found was that with increasing concentrations of BPA or estradiol, there was an increase in expression of this binding protein. And this was blocked with ICI, an estrogen receptor antagonist. But it does not occur in the tumorigenic cells. And so this is exciting because it's showing us that maybe this is the mechanism whereby estrogen might be doing this within the prostate epithelial cells. To now tie those two events together to show evidence that MBD2 actually directly regulates the methylation status of phosphodiesterase 4, Winnie more recently used these same cell culture models, looked at PDE4 expression in the presence of sense RNA transcripts or antisense RNA transcripts to this molecule. And with, of course, increasing BPA or estradiol, you see increased expression of PDE4. But when you use antisense mRNA, you totally block the increased expression of PDE4 when you block MBD2 gene activity. And this is directly correlated with changes, as one would predict, in the methylation status of that gene. So normally, for example, bisphenol A would be resulting in hypomethylation of these genes. With antisense to MBD2, it blocks that process. So in the normal cells where BPA and estrogen hypomethylate this gene and increase gene expression, blockade of this molecule with antisense RNA directly blocks this effect. So we conclude that perhaps BPA and E2-induced increases in this expression are mediated by increased transcription of this gene. I want to just show you a little bit of data that this may be occurring in the human phosphodiesterase 4 gene as well. So this is in normal prostate epithelial cells. We likewise see a pressing the wrong buttons. A CPG island in the promoter region of that gene. In normal cells, there appears to be a hypermethylated region that can be demethylated, resulting in increased expression of that gene in human epithelial cells. And we also looked at some prostate cancer specimens. 3 patients are shown here where there was non-neoplastic tissue removed by laser capture and tumor tissue from the same patients. And the non-neoplastic regions, the phosphodiesterase 4 gene was primarily methylated, whereas in the tumor cells it was primarily unmethylated, showing that there may be some very similar parallels to what we're finding in our animals. There's one last gene I'm going to show you with one last slide, just to show you that we are moving on to other candidates as well. And now we're looking at this gene HPCal, which stimulates cyclic AMP production within cells. So it's connected in the same signaling pathway, but it has opposite effects. And we looked at this particular gene, its methylation status by MSPCR in our noble and AIT cells. For the most part, it's unmethylated, has high expression patterns, but in the tumorigenic cells, it becomes it starts becoming methylated. So we start seeing some methylation of this gene associated with decreased expression of this gene in the tumorigenic cells. And it appears to be methylation regulated because that effect can be reversed with treatment with a demethylating agent. So why are we looking at this? Well, when we did our screens, we found a very interesting phenotype with regards to this particular gene. Nothing at early days, but at day 200 of life. Normally, this gene is entirely unmethylated, and there's high expression of this gene. But if they've been exposed neonatally to BPA, it was hypermethylated, and it was not expressed. All right? As those animals started developing preneoplastic lesions due to adult T+E, expression was reduced in all the treatment groups. And so it's primarily an unmethylated gene, and it's expressed in normal tissues. And early BPA exposure results in its methylation and reduced expression. So our summary and conclusions right now from what I've shown you is that the prostate epigenome is altered by early life exposure to estradiol or BPA. We've identified a number of candidates that show differential methylation. We're seeing some gene-specific changes that vary with regards to dose and exposure. We see other ones as well, but we have convincing evidence right now for PDE4 and HPCal2 genes involved in regulating cyclic AMP levels within the cells. And so we suggest that perhaps these and other candidate genes may alter signal transduction that contributes to prostate carcinogenesis with aging. So our overall working model that we have right now is that normally prostate gland develops, as I showed you at the beginning of my talk. We have progenitor cells which cytodifferentiate, become functionally differentiated secretory epithelium of the prostate gland. And we propose that if there's exposures to abnormal levels of natural estradiol or to exogenous compounds with estrogenic activity, such as BPA, we can initiate epigenetic changes in these cells. We can have epigenetic organization, and they will alter or imprint or reprogram progenitor cells within the prostate. This results in differentiation defects as the animals age, and with additional adult insults, this can predispose to pin lesions and prostate carcinoma as the animals age. So I would like to acknowledge these folks in my laboratory, particularly Jessica Belmonte, who is involved in the BPA work, and these others in the estradiol work, from Shuk-Mei Ho's laboratory at the University of Cincinnati, in particular Winnie Tang, who's now a research assistant professor in Dr. Ho's department. Thank you very much. I'll take any questions.
Thank you. Thank you very much. And,
no, that's
This is a wonderful talk. Now we have time for questions. I actually already received a set of questions from Dr. Kim Hooper at DTSC several days ago after our lecture announcement. And Dr. Prins, could you address those questions first?
Okay, so the question was, is there any evidence that diet, so eating high levels of tofu with its phytoestrogens or pomegranate juice or other foods with high levels of antioxidants affects the risks of enlarged prostate or initiates or promotes prostate cancer, Gleason score, blah, blah, blah. And yeah, there is. And actually, it seems to be protective. And so there seems to be a divergence of estrogenic effects. Those would be estrogenic exposures in the adult. So we're not talking then about developmental estrogenic exposures. We're talking about estrogenic exposures in the adult. And some of these phytoestrogens and compounds actually are showing a preferential liganding to estrogen receptor beta within the prostate epithelial cells. And there's evidence that estrogen receptor beta may actually be an anti-proliferative steroid receptor. And it is involved in repressing growth of the epithelium. It's a very complicated story because its levels change in cancer in interesting ways, but that's what the suspected mechanism is right now, that some of the phytoestrogens might actually be protective against prostate disease in adulthood by activating an anti-proliferative pathway. And that's very different from the developmental exposures. Those developmental exposures we showed with the ER-alpha and ER-beta knockout animals are actually mediated through ER-alpha, not through ER-beta. During the developmental period, it's ER-alpha in the stroma that appears to be doing these effects.
What about phytoestrogen in pregnancy?
Yeah, and so that's very controversial. There is some evidence that that can be detrimental. Yeah, one needs to be careful with that. There certainly is a lot of evidence with that now in the breast, but there is also some evidence with that in the prostate that's not as well investigated as the breast. But that deserves caution as well.
I don't know if you can actually answer this, but I'm trying to correlate if there is something like a BPA syndrome, if you will, given that this other group has found neuronal circuitry being altered.
Yeah.
So would there be a subpopulation who maybe would be susceptible to, via the two-hit theory, come up with prostate cancer? Would that population necessarily also show, symptoms or signs of this altered neuronal circuitry? Or should we be looking at that subpopulation for that? What are your thoughts?
Maybe we should be looking at it to tie it all together. I mean, what we're seeing in the prostate may be very tissue-specific because of the genes that are expressed there. What we've looked at so far has been these epigenetic marks in prostate cells, and they're going to be very different in other cell types. So it's going to depend on, for example, whether they can respond to a second hit of estrogen and alter neuronal circuitry. Is that what you're getting at? Would the second hit apply to other systems as well?
No, what would the second hit do? First of all, I mean, it's sort of like two things. If, in fact, to confirm that the effects seen are actually BPA-driven and not other confounding factors involved.
In humans.
In humans.
Yeah.
This is finally, we probably will use animal models and then make the extrapolation later.
Yes.
But the
It's
question...
going to be very difficult to dissect this out for human because of the confounding variables, because people are exposed to so many agents at the same time, that the ability to dissect that out is going to be very, very difficult. That said, as well, the disease we're looking at is an aging-associated disease. And so to correlate what's going on with a man when he's 50 to something that happened during their developmental period, it's going to take a long time to begin with. And that's going to be very difficult to do because of all different lifestyles throughout life and exposures throughout life. So that's a real hard one. We need to find surrogate markers in the meanwhile that might suggest other predisposures to prostate disease. But back to a syndrome. You know, I don't want to go on record as saying, yeah, there is, and so I certainly will not, but there are clearly effects of bisphenol A outside the prostate. There are effects on puberty, there are effects in the female, there are effects on brain, there are effects on a number of other systems. And so what drives that is there a person or an animal that is susceptible, more susceptible to all of those abnormalities versus another one that isn't? I don't think has been looked at in an individual person or animal. Usually our scientists are all,
right.
You know, I'm prostacentric, and so I'm sitting there looking at the prostate, and somebody else is looking at the neural system. We haven't looked in our animals whether they're carrying the same thing. So maybe this is something that's worth evaluating.
I was wondering, like, if maybe the neurobehavioral guys could share their tissues with you, and you could take a look at it.
Yeah, that would be interesting. Yeah, to see with regards to susceptibility strains. What drives susceptibility is very complicated. So something very important to think about, though, going forward. Very important. Thank you.
I'm curious, what's the rationale for using the combination of T and E when hitting the adult instead of just E?
Oh, that's basic endocrinology. Okay. If you give an animal or a human estradiol, you'll shut down the pituitary. You'll feed back on the pituitary. The pituitary will stop producing luteinizing hormone to drive production of testosterone. And so it would be referred to as a chemical castration. You would be removing testosterone by giving chemicals. In fact, this was the original method for treating prostate cancer devised by Charlie Huggins, which he won the Nobel Prize for, was giving elevated estradiol to these men, give them a chemical castration to shut down their testosterone. And their prostates would regress, and their prostate cancer would regress for a time anyway, before it became androgen independent. And so we clamp the animals with physiologic levels of T, because we don't want the prostate to just regress.
I have a question about your two-hit animal model.
Yes.
And I couldn't help to notice after adult estrogen insult, the BPA treatment group have significantly increase on PIN number.
Yes.
Yeah, and compared to the low estrogen treatment group,
right.
Which didn't change at all. So does that means BPA can work through other mode of action besides the estrogen?
That's an excellent question.
Yeah, besides the classical estrogen.
So there's more. Yes, first
pathway.
of all, yeah, that's an excellent question, and so let me address that a little bit. Estradiol itself will affect cells through, if we're talking about estrogen receptors, it's going to affect it through genomic estrogen receptors, the nuclear molecules that activate gene transcription. Mm-hmm. And it's also going to affect it through estradiol binding to membrane-bound estrogen receptors, which we now know. BPA has a very low affinity, very, very low affinity for nuclear estrogen receptors, but it has about an equivalent affinity for membrane-associated. And so we might be finding there differences with regards to which pathway is being activated by estrogen, by BPA, if in fact it is an estrogen receptor pathway. The data that Winnie now has with the normal and AIT cells, where she can block the effects with ICI, suggests that it is a receptor-mediated phenomenon with regards to BPA that we were showing you on those cells. But there also could be non-ER-mediated effects of BPA. We can't rule that out. But right now we think that there's dose and chemical different responses, most likely primarily or to a large extent related to the different liganding and activational capacities for the different receptor molecules.
Okay, yeah, sounds interesting. And can you also explain how come the high-dose estrogen treatment group has a similar effect compared to BPA, but not low-dose estrogen treatment group?
when they're at high doses neonatally, a number of systems are immediately perturbed, where what I would refer to as structural reorganization. And that's what I really focused most of my research on during my research career, is looking at perturbations in some of the steroid receptors expressed during development and the developmental genes that regulate development. And they're perturbed in expression, and they drive the development of a very abnormal looking gland. Mm-hmm. The low doses don't do that. Nice. And so you're going to get differences depending upon doses.
Thank you. Just last one. I noticed that there's a last gene you mentioned, HPCAL1.
HPCAL1.
Yeah. Is that one BPA-specific gene? Only responds to the BPA
that's
treatment?
That's all we found so far in our data. Oh, is that the only one that was BPA-specific?
I don't know. I see you show that in the
slide. It was BPA-specific. It's not the only one that was BPA-specific, but only BPA seemed to affect that, not estradiol, in our tissues.
So that could be a possibility. A group of the gene can only influence by BPA treatment.
That's what we would suggest.
Not by the estrogen. Thank you. Yeah, that's a wonderful talk.
So we believe that there'll be differential effects. Our goal is to find a cohort of these genes that can serve to be fingerprints for maybe different exposures during development. Wouldn't that be wonderful? And you could say, well, you seem to be predisposed because your fingerprint suggests that you had more alterations in the genes that suggested BPA exposure versus elevated maternal estradiol. That would be wonderful.
I have two questions. One is, have you done some transgenerational studies to see if some of these effects then go on to express themselves in future generations? And the other is, what is the reason, what causes elevated estradiol levels during pregnancy? Abnormal. I mean, their lifestyle? Is there something that is it genetic? Kind of just, if you could.
Okay, I can answer the first question really fast, and that's no. We have not looked at transgenerational effects. That's a huge amount of work beyond the huge amount of work we're already doing. And so we've decided to drill down more specifically in the first generation. And where our real focus is going now is to tie these changes into carcinogenesis. Because right now these are observational related events. And we have to show now that these genes, such as PDE4, are actually involved in carcinogenesis and these other genes. And that's what we're very active doing. So we're being very prostate specific there. I would love to have a bigger lab and more money to look at transgenerational effects. But we haven't begun to do that, but it would be wonderful to do that. So that's a good question. What regulates different estradiol levels? Well, multiple things. They believe in the data from Ron Ross's group from University of Southern California would suggest that that's due to polymorphisms in estrogen metabolizing enzymes that result in higher levels versus lower levels in different populations. And so that's been shown quite nicely for the African American population versus the Caucasian. So that's one way. Another would be to differential clearance. Another one could be to differential levels of production, different sensitivities to the hormones that are producing the estrogens. There's a lot of diseases that affect estradiol levels in the mother. So preeclampsia affects it. Hydrocephaly actually drives massive amounts of estradiol levels. That's something I came across very early on when I looked at this literature decades ago, was that good pathologists can diagnose hydrocephaly by looking at a prostate cross-section because there's such massive squamous metaplasia of the epithelium of the prostate because of the elevated estradiol in those fetuses. So different diseases will affect your estrogen levels. And so maybe there's more explanation. Jimmy Spiro probably could give you more answers. But there's large variability in the human population.
This question.
Oh, and diet. You asked, can diet do that? And I would very much suspect that it does. Yes. You can eat compounds that are higher in estrogenic agents. There's some evidence that maybe eating diets high in phytoestrogens might not be the best thing, although we don't know right now. I wouldn't run out and do it because it's not been proven that it's negative, but there's some data which suggests that it could be. But certainly your dietary levels of various compounds that you eat can modulate your serum levels of hormones at a variety of levels in the hormonal axis. Certainly you can eat estrogenic-like compounds. Also, the amount of methyl donors that you have in your diet through folate is going to affect gene expression through modifying epigenetics. And so there's a lot of ways that this could take place through diet.
Okay, this question comes from the internet from one of our toxicologists at our Oakland office, Dr. David Morey. His question is, would obesity in older men cause higher estradiol levels and affect prostate cancer risk?
Yes, yes. There's direct evidence for that.
Yeah.
Particularly in the African American population, it turns out.
To follow up on the comments you made earlier with respect to with respect to variability in hormonal levels in the human population, yeah. I was very struck by the magnitude of the difference between African Americans and Caucasian population. And as we know, African American populations are genetically very diverse, so I suppose if you looked at African populations, that difference might be even greater. So given the magnitude of the differences, wouldn't you think that would overwhelm any differences that you might get from environmental estrogens?
Yeah. I want to say a few things about that. In no way do I mean to infer that maternal estrogen levels are the cause for the differences in prostate cancer rates between the African American and Caucasian that are known. There's a lot of evidence at a lot of different levels of from everything from lifestyle to genetics to all different kinds of things looking at these different populations. This is one observation, that they do have elevated estradiol levels during pregnancy. African Americans have the highest rate of prostate cancer in the world, so it's much higher than the prostate cancer rate in African men. All right, and so we understand that it's a very heterogeneous population. So what's driving that is not fully well understood. To a large extent, it could be lifestyle. The lifestyle in the United States, the environment, is not necessarily conducive to prostate health. So we know that Asians who come to the United States, Asians in Asia have the lowest rate of prostate cancer worldwide. But within a generation of moving to the United States, their rates of prostate cancer begin to increase. And so, and that's without interbreeding with other races. And so there certainly is an environmental impact and a lifestyle impact that is contributing to that. And it's very complicated. Does that in any way answer your question?
Well, sure, but I guess what I was thinking is that it just seems like mom's estrogen levels would have by far the greatest impact on the developing fetus.
On the developing prostate, perhaps, yes, yes. But there's a lot of other causation for prostate cancer besides what's programmed during development.
Sure.
Yeah. Changes in mutation rates, androgen receptor polymorphisms, all different types of things. Not polymorphisms, differences in repeat length, in the CAG repeat length, and differences in the androgen receptor activational capacity. There's a lot of differences being uncovered, certainly dietary. Certainly dietary.
Very nice, Gail. What I'm wondering about is, we have these big differences in the frequency of non-metabolizers, and for example, in the African American population, you find a high incidence of individuals that have Gilbert's syndrome, which are deficient for the UDP gluconeotransferase enzymes, and which is some of those, not all of the different alleles, but some of those are involved in metabolizing BPA, yeah. And yet we're using animal models that are all high metabolizers in the different kinds of
My understanding
screening
is that
assays.
the animals actually metabolize, the rodents metabolize at a lower rate than humans.
Right, but all I'm getting at is I'm not sure that we really looked at that association in the human population.
No, that has not been looked at. Certainly in the human population, that's the different polymorphisms and how that might contribute to effects has not been well studied in the animal literature either, that I'm aware of. But I don't know all the literature, certainly.
But what about polymorphisms in the epigenetic mechanisms that may be involved in gene silencing or hypomethylation? Yeah. Because that's a whole another area that we really haven't considered the effects of these endocrine agents on.
Absolutely, and I'm not aware that anybody's even doing that with regards to those metabolizing enzymes. There's some real interesting data that with bisphenol A exposure during life, how that can be shifted and reversed with folate exposure, dietary folate during that period of time. And we're looking at that in our prostate tissue right now as well with regards to BPA. But you're talking more specifically about polymorphisms in the enzymes that metabolize it, and I'm just not aware that that's been thoroughly investigated in relation to negative effects of BPA. They exist. Polymorphisms exist. Yeah.
Okay.
I have two questions, Gail. The first one is you have two hit models, the neonatal exposure, then adult exposure. Lots of people in the academic labs use the one-hit model, just the developmental exposure. But lots of industrial studies on bisphenol A, you have large scale, 2-generation, 3-generation studies. The animals receive bisphenol A through their lifetime, I mean, before preconception until the aged period.
Correct.
What are those two exposure scenarios have different results? I mean, the controversial issue on bisphenol, one of them is why those large-scale 2-3-generation studies did not observe.
Because they were seeing BPA throughout life. So theoretically, you're saying they saw their second hit. They saw a whole lifetime hit. Well, that was to BPA. It wasn't to estradiol. It wasn't to elevated estradiol. And that might be one explanation. Strain differences may explain. There's a lot of investigation into why some groups have seen BPA effects and others haven't seen BPA effects after different periods of time. I'm very familiar with all that literature. But if you want to ask a question specifically about lifetime exposure to BPA being the second hit, we didn't look at a second hit of BPA. We looked at a second hit of elevated estradiol. So I think that would be the difference. And what we were trying to do specifically is, so our evidence suggested that BPA by itself would not necessarily be carcinogenic. It would not be sufficient to drive carcinogenesis by itself of the prostate. But it might initiate some changes that other exposures then might promote and add on to those specific changes. If you want to look at the initiation promotion model of carcinogenesis. So we're looking at it more with an eye towards that. And so our second hit, our second exposure, we modeled to be something that might take place in an adult male, which is relative increases in estradiol levels.
Interesting. Thank you. My second question, back to bisphenol A again. And you look at the NTP draft brief. The number one is the neurobehavioral effect.
Yes.
And you mentioned the MECP2. In one lecture we had by Dr. Lassell from UC Davis, he looked at the role of MECP2 in the neurodevelopment, the autistic process. So I'm curious to know, have you looked at the changes in MECP2 by either estradiol or BPA?
In that last study that I showed you that Dr. Tang did, she did look at MECP2. And there were effects of MECP2. They weren't estrogen receptor antagonized with ICI. It was different, and it was in response to, I believe, estradiol and not BPA. So there were some differential effects going on there. It became more complicated. I will say that MBD2 is also, if I have this correct, involved, is a binding partner in the MECP2 complex that sits down on genes, that they are connected molecules that one sits down and recruits the other one. So there may be things going on at that level. We were just looking at changes in gene expression and methylation status. And we did see some, but it became more complicated, so I didn't show that to you now.
That's interesting because that could be one set of molecules that hook up all the endpoints, the neural effect, the prostate, maybe something else.
Yeah, it could be. Yeah. So I'll have to look that up. I didn't know that there was a commonality there.
Yeah, thank you. Thank you. Yeah. Any more questions from the room? Yeah. Let me check the email. Maybe that will wrap this up. We do have one.
All right.
Well, in the meantime, I have one question. A lot of pharmacokinetic studies on bisphenol A suggest that the oral route is the most important route. Mm-hmm. I'm not sure I caught in your talk what route you used, and if it wasn't the oral route, which do you feel is the most relevant to human exposures?
Okay, thank you very much for bringing that up. That's what my comments to the draft panel on NTP were, exactly on that whole issue. We gave our neonatal animals subcutaneous injections of bisphenol A, not the oral route. And so that has been criticized. It was criticized by the initial panel as a weakness. The initial subcommittee saw that as a weakness in experimental design. And, okay, so why do we do that, and why might that matter or not matter? And we do it because we can control exactly the level of BPA that it's getting into each of those animals. And you can't do that with oral exposure to mother and then feeding. You could do it by gavage, but yeah, we didn't do gavage. That's a stressor. I guess subcutaneous injections are as well. But more of the major issue here is that the enzymes that Jimmy was talking about, the UDPGT enzymes that metabolize BPA, aren't expressed in the neonatal rat liver. And there is a paper out now showing in the mice, at least, that if you give oral exposure versus subcutaneous exposure, it results in the exact same level in the serum. We are in the midst of pharmacokinetic analysis in our Sprague-Dawley rats, repeating that exactly. And so we've already done a very extensive study on the male and female rats at separate days of life neonatally. And we're now looking at the serum levels of BPA and all their metabolites using MSMS to determine that. And we hope by the end of the year to have that data published. So we'll be addressing that directly. But the data that's coming out suggests that this might not be as big of a player in the neonatal period. Certainly in the adult period, it's going to make the world of difference. But in the neonatal period, where that's not being metabolized anyway, we didn't feel that it was sufficient to change our protocol. But it was a very important point that needed to be addressed. Route of exposure is going to be very, very critical. And I have a problem with giving it to the mothers and then getting it through through the milk, through feeding that way, because there's so much variability then from animal to animal in actual exposure level and intake that becomes very hard to control, leading to a lot of variability in data. And when you have variability in data, you don't get statistical effects. And so I don't like that impreciseness of that particular approach. That's my bias.
Thank you very much, Dr. Prins. Thank you very much for flying all the way to the summer Sacramento, then fly back 2 o'clock this afternoon. And thank everybody for participating.
Thank you.
And I will give this mic to June to wrap up our current series.
I just wanted to mention a few things. First of all, Dr. Prins, that's an excellent wrap-up presentation to our series.
Thank you.
It was very, very good. We have been privileged to hear from many of the experts in the area for this series, and so it's another great addition to what
We
we've
did
been
have a
doing.
great series.
We did.
Yes.
Yes. We had people from all over the world listening in, and may have them as well. So I want to acknowledge that. I also want to acknowledge the work that Ling Hong has done on this. The idea for this actually came about during a conversation that I had with Ling Hong last year, and he has done a mammoth amount of work to contact the individuals, to set up their travel schedules, to arrange that we here at Cal EPA, as well as the web audience, could hear from such a distinguished group of speakers. So I do want to acknowledge Ling Hong. We're in the planning processes for a future distinguished lecture series, so we'll let you know when we kind of formulate that. So I want to thank the audience, the individuals who have participated, Ling Hong, the staff, George, the people who are on the web, and the distinguished speakers for a really great seminar series, which are available. The talks are available on our web. So thank you very much.